So, how do you fix a disease that's written into your DNA? Do you add a brand new, correct page to your body's instruction manual? Or do you find the exact typo on the original page and just rewrite it? We're about to dive into two absolutely revolutionary technologies that do just that: gene therapy and gene editing. Now, they sound pretty similar, right? But the way they work is fundamentally different. And, getting that difference is really the key to understanding where medicine is headed. So let's break it all down. All right, here's the plan. We're going to start with the basics, our genetic blueprint. Then we'll get into gene therapy, which is all about adding a new gene. After that, we'll tackle gene editing, rewriting the code itself. Then we'll put them side by side to see how they stack up, look at the incredible promise, but also the serious challenges, and finally, we'll talk about what this all means for the future of medicine.
Okay, so before we can even begin to talk about fixing our genes, we really need to get a handle on what they are and what they do. Just think of your DNA as the ultimate instruction manual, the blueprint for building and running a human being. But what happens if there's a typo in that manual?
Now take a look at this. This visual really shows you all the layers of our genetic code. Let's start on the left with a single human cell. You see, inside almost every one of your cells, there's a nucleus, and that's where our chromosomes live. Now, if you could zoom way in on one of those chromosomes, you'd see it's just a super long, tightly coiled strand of that famous double helix we all know, DNA.
So what even is a gene? Well, it's just a specific chunk of that DNA. A great way to think about it is like a single recipe in a giant cookbook. Each recipe, or gene, has the instructions to make one very specific thing your body needs. And that thing is usually a protein. And let me tell you, proteins are the absolute workhorses of our cells. They do, well, everything, from carrying oxygen around your body to fighting off infections. So that brings us to the most critical question. If the recipe is perfect, you get a perfect protein, and it does its job exactly right. But what happens when there's a typo in that genetic recipe? What if just one single letter is out of place in our 3 billion letter code? And here we have a perfect and honestly devastating real-world example. Let's look at sickle cell disease. So in the gene for hemoglobin, that's the protein that carries oxygen in our blood, the DNA sequence is supposed to read GAG. This recipe makes an amino acid called glutamic acid, and everything just works. You get nice, round, healthy red blood cells. But in people with sickle cell, one single letter gets swapped. That GAG becomes GTG. This tiny change swaps out glutamic acid for a different amino acid called valine. The result? It's catastrophic. The hemoglobin protein starts clumping together, forcing red blood cells into that rigid sickle shape you see here. Those cells can block blood flow, causing unbelievable pain and organ damage. All of that from one single typo.
So you're faced with a faulty gene. What was the first strategy that scientists came up with? Well, it's called gene therapy. And the big idea here isn't to fix the typo in the original recipe. Instead, it's just to give the cell a brand new, correct copy of that recipe to work from. The official definition here really gets to the heart of it. It's all about delivering genetic material to compensate for a problem. Notice that word, compensate. The faulty gene isn't taken out, it's still there. What happens is a new, working version is added alongside it. The hope is that the cell will just start reading the new, correct instructions and ignore the old, broken ones. It's kind of like sticking a corrected recipe card into your cookbook without bothering to tear out the old one. But that, of course, leads to a massive technical problem. I mean, how do you actually get this new piece of DNA inside the trillions of target cells in a person's body? You can't just inject it and hope for the best. You need some kind of delivery vehicle.
And this is where scientists got incredibly clever. They decided to repurpose nature's own delivery experts, viruses. Let's walk through how this works for hemophilia, a disease where people can't make a protein for blood clotting. First, scientists build a correct, functional copy of that clotting gene in the lab. Then, they take a virus, strip out all the nasty, disease-causing parts, and they package the new, healthy gene inside it. This modified, harmless virus now acts like a delivery truck, or what they call a viral vector. When this vector gets into the patient, it does what viruses do best. It finds its way into the cells and drops off its genetic cargo. The new gene can then start giving the cells the right instructions to make that missing clotting protein. Pretty smart, right?
Now, there are basically two ways they can make this delivery. The first one is called in vivo, which just means in the living. This is where that viral vector is injected straight into the patient, kind of like in the hemophilia example, and it has to find its way to the right cells all on its own, inside the body.
The second method is ex vivo, which means outside the living. For this one, doctors actually remove the patient's cells, take them to a lab, use the vector to add the new gene there, and then infuse those freshly modified cells back into the patient.
Okay, now let's switch gears to our second approach. If gene therapy is like adding a new recipe card, while gene editing is like taking a pen and directly correcting the typo on the original page, it's a much more direct and potentially a much more permanent fix.
You know, the key words in this definition are everything, permanent and at a specific location. See, unlike gene therapy, which just kind of adds a gene somewhere in the cell, gene editing aims to find the exact spot in our 3 billion letters of DNA that has the error and fix it right there on the spot. It can cut out a bad sequence, correct it, or even add a missing piece back in.
And the technology that really made this precise kind of editing a reality is called CRISPR-Cas9. You might have heard it described as a pair of molecular scissors, but honestly, it's more accurate to think of it as the find and replace function in a word processor, but for our actual genetic code. It has a guide molecule that lets it find the exact DNA sequence it's looking for, and then a cutting tool that snips the DNA at that precise spot. It's a total game changer.
So, what does this look like in the real world? Let's go back to sickle cell disease. First, the patient gets medication to push their blood stem cells. These are the factory cells that make all your other blood cells out of the bone marrow and into the bloodstream. Second, a process called apheresis, which is sort of like a really sophisticated blood filter, collects these specific stem cells. Third, and this is where the magic happens, these cells go to a lab. The CRISPR-Cas9 tool is sent in, and it meticulously finds that single GTG typo and rewrites it back to the correct GAG. Fourth, and this part is intense, the patient gets chemotherapy to wipe out their remaining faulty bone marrow. This makes space for the new healthy cells. And finally, those newly edited healthy stem cells are infused back into the patient. From there, they set up a new factory that can produce healthy, round red blood cells for the rest of the patient's life.
Okay, so we've looked at both approaches on their own. Now it's time to put them side by side to really nail down the differences between gene therapy and gene editing.
So here it is, the simplest way to think about it. Gene therapy adds a new working gene to make up for a faulty one. The bad gene is still there. The analogy's perfect. You're just sticking a new recipe card into the cookbook. Gene editing, though, it directly modifies the original DNA. It finds the error and it fixes it. It's like taking out a pen to correct that typo on the original recipe page itself.
This table breaks it down even more clearly. Let's look at the goal. Therapy is trying to compensate, while editing is trying to permanently correct. The method, therapy adds a gene. Editing alters the original DNA. The tool, therapy usually uses a viral vector as a delivery truck, while editing uses those molecular scissors like CRISPR. And this last point is so important, permanence. Gene therapy can actually be temporary because that new gene might not get passed on when cells divide. But a gene edit, that's a permanent change to the chromosome. When that cell divides, all of its daughter cells are going to inherit that corrected gene.
Now look, these technologies are not science fiction. They are being used to save lives right now, and their promise is just incredible. But they also come with some massive challenges and raise some really profound questions that we're only just beginning to deal with.
And these aren't just theories. They are real, life-changing treatments. Gene therapy is being used to treat hemophilia. Gene editing, as we've talked about, is now a functional cure for some people with sickle cell. And there are even gene therapies that can restore sight in certain kinds of inherited blindness by delivering a correct gene right to the cells of the retina. These are amazing success stories.
But it is not easy. There are still huge hurdles to get over. Just getting the delivery right is a major challenge, especially for those in vivo therapies where you inject the vector directly. How do you make sure it goes to the right cells and absolutely nowhere else? The procedures themselves can be incredibly grueling. I mean, think about the chemotherapy needed for the sickle cell treatment. And then there's the cost. These are highly personalized, complex treatments, and the price tags can run into the millions of dollars per person, which raises some really tough questions about who gets access to these cures. And that really brings us to the biggest question of all. Let's go back to that definition of gene editing, the permanent removal, correction, or addition of our own genetic code. For all of human history, our DNA was something we just inherited. Now it's something we can edit. This is a monumental shift, and we have to ask ourselves, what does this truly mean for the future of our species? So where does this all lead? Well, what we're witnessing is a fundamental paradigm shift in medicine. For centuries, medicine has been about managing the symptoms of a disease. These new technologies, they offer the chance to go straight to the source of the problem, our DNA,
and fix it for good.
So let's bring it all home. We started this journey with gene therapy, which was that incredible first step. It showed us it was even possible to add a new set of instructions to the body. But now, with tools like CRISPR, we are in a whole new era, one where we can actually rewrite the original manual itself, correcting the errors for good.
And so, this is where we end. For the first time ever, the Code of Life isn't just a text we can read, but one we can write. The power to cure these devastating genetic diseases is finally within our grasp. But with that power comes an immense responsibility. The technology is here. Our genetic code is now editable. So the only real question left is, what should we write next?